Welcome everyone to our Spotlight Theater. My name is Luigi. I will be the chair of this session where we're going to hear from Professor Harold Cremer from the University of Marseille and how our imaging product has helped him advance in his research on brain cancer. But before handing over to Harold, I wanted to spend a few words on Miltenyi Biotech in case you don't know us and what do we do with a particular focus on our imaging and spatial biology portfolio. Now, Miltenyi is a biotech company founded in 1989 in Germany that has been rapidly growing ever since. To this date, we have subsidiaries in 23 countries, including North America, and we are in general represented in 73 countries. Since its foundation, innovation has been the main driving force of the company, and that's a statement that still holds true today. As you can see, indeed, around a fourth of our employees are still working in research and development. And another peculiar thing about Miltenyi is that when I mention research and development, I'm not referring just to the research that goes into the products, but we also perform actual scientific research. So we have proper research group that use our instrument to produce scientific data, which is a win-win because not only we are helping the field moving forward, the cancer research field, but it also makes us not only experts of the technology, but also on the applicative side. So we are day by day helping scientists to achieve and have successful experiments as they were planning to. Now, regarding our products, our portfolio is very broad. I'm sure that most of you know us from either one or the other side of the products portfolio. We really go from bench to bed, starting from products for cell preparation, sample preparation, and cell isolation, all the way up to automated, highly automated actually, cell manufacturing platform. But again, for the sake of time today, we are just going to focus on the imaging and spatial biology portfolio. Currently, this is constituted by two platforms, the ultramicroscope platform for 3D imaging of whole large samples or tissue, and the Maxima platform, our system for spatial biology applications. I would like to have a quick introduction for each of these platforms, starting for the ultramicroscope and 3D imaging. First of all, why should you be interested in 3D imaging at all? The answer here is quite easy because the world we live in is in 3D, so it just makes sense to analyze samples and tissues in their natural context whenever possible. I brought here an example on what 3D imaging can give you more compared to more traditional approaches. What you see here, left and right, is the exact same sample. We're looking at the pancreatic xenograft tumor. Three markers, you have vasculature in green, CAR T cells that these tumors were treated with in blue, it's a CD3 staining, and in red you just have autofluorescence, which is typically used to get an overview of your sample. As you can see, the amount of information that you get out of the 3D render, out of the 3D imaging session, is a lot more. If you focus, for example, for the vasculature, that's typically a very good example on why 3D imaging is needed. You can directly reconstruct completely the vasculature out of your tumor, whereas if you look in the standard 2D sections where we simply just cut slices out of the tumor and image them, most of these vessels will just look like points, so it's going to be hard to reconstruct the entire structure as it is originally is, on top of being a lot more time-consuming and requiring hands-on time. Now, this is the why you should be interested in 3D imaging. How we do that is shown in this slide. Very briefly, we have our full 3D imaging workflow. Point number one, you start with your tissue, as I mentioned, as a whole. You no need to cut, no need to modify it structurally, it's simply fixed. You then proceed with the antibody staining. In principle, there's no difference compared to a classic immunohistochemistry workflow on slides. The only difference is that staining times are going to be a little bit prolonged because you need to ensure that the antibody penetrates deeply into your tissues. The next thing you gotta do, and this is a key step in the 3D imaging workflow, is tissue clearing. Tissue clearing protocols are protocols that make your sample optically cleared, in other way transparent. Hence, you are able to look through it. And that's really the key step that allows for this imaging of your samples as a whole without the need of sectioning them. Step number 3 is, of course, imaging. So now you have your sample, it's been stained, it's been made transparent, and can go directly into the microscope. What we recommend, what we think is the best system to perform 3D imaging is light sheet microscopes, because they're very gentle on the samples, so your fluorophores are going to be very happy for the whole imaging time. And they're also very fast, especially compared to more traditional epifluorescent microscopes like a confocal. Now you have your nice 3D overview of your sample. Of course, you might want to do some processing of those data, some analysis, or handling in general. So software is also a key component of every 3D imaging workflow. This is actually general. It does not apply only to our products. Anyone who should be looking into 3D imaging should perform all these steps. But when it comes to Miltenyi, in our company, workflow is a word that you might hear a lot, because for each of our products portfolio, we don't offer just the instrument, such as the light sheet microscope, but we really offer everything that is needed to run the workflow, including reagents, consumables, softwares, as well as scientific support at each of these steps. If we look at the ultramicroscope specific workflow, of course, we have our very fast and fully automated 3D imaging systems. We have 2 platforms at the moment, the Blaze and the Coros. They're very similar. The difference is that the Coros gives you even more imaging range if you are interested in those very large sample or even large organisms. For example, a whole mouse could be imaged with the Coros. We have clearing kits, in particular 2, the Max clearing kits and the Max deep clearing kit, which, as the name suggests, is the one that we recommend for those larger samples where it's a bit more challenging to get deep antibody penetration, hence the name. Again, we don't just offer the kits, but we also validate them and have freely available protocols for different tissue types, which you can have a look even right now on the fly on our website. We have a number of tissues, of organs that have been tested, and you can find really step-by-step protocol involving our clearing kits for those specific tissues. Similar story for the antibodies. We have our own portfolio of antibodies validated for 3D imaging. They are all primary conjugate antibodies. We have, at the moment, 4 fluorophores available. And also that if you have a look on our website, you will find the full list, as well as a lot of additional information about the validations, some example imaging, so that you know what to expect from your 3D imaging experiments. We have a software, MaxIQView 3D software, dedicated for image processing in case you want to even enhance further the already pretty nice images that you get out of the microscope. And of course, scientific and technical support at each of the steps of the workflow. So if you're encountering any troubles or have any questions, you can always reach out to us and we'll be happy to help. Now, to conclude the 3D part, I just wanted to bring some examples of what you could expect from our workflow. It's just really the instrument and the whole workflow in general, it is so flexible that the limit is your imagination. Whatever samples you are looking or interested in, you probably would be able to image it in our microscopes. Our imaging chamber are also very large, which ensure that whatever samples you are interested in, you would be able to fit it in there. But another way, which we are very excited on how to use all this space, is to have a lot of these small samples. For example, on the top right, you see 48 organoids that have been imaged sequentially under our system. And also in that case, we have a dedicated workflow for this application. We have protocols available online, which I really encourage you to have a look at. Not only pretty pictures, but also meaningful research. As of this year, we passed around 1,000 publications where the ultramicroscope technology has been cited. A lot of papers, a lot of high impact factor, and in some cases, our pictures even made some pretty nice covers of high impact journals, as you can see in this example. Moving on to the next platform, the Maxima. Maxima is a spatial biology platform, so it allows you to do deep phenotyping, get as many markers as you need out of your tissue slide, as opposed to a standard, for example, 5-plex immunofluorescence assay as shown on the left side. So you're really able to characterize your tissues with whatever markers you are interested in. Again, on the how it does this, it's via this mix technology. Mix stands for Maxima Imaging Cyclic Staining, and as the name suggests, it consists of cycles, 3-step cycles that are repeated over and over. The specific steps, as you can see in the animation playing, are stain. You stain for 3 markers for each cycle. You image them, erase the 3 markers, and then move on to the next cycle. And this can be repeated as many times as you want. When I talk about markers, typically we refer to protein markers. However, it was at this very same event last year, AACR Spotlight Theater 2024, where we presented for the first time our RNA Sky technology, which allows for true multi-omics, single-slide multi-omics. So now what you would be able to do is to detect RNAi first in a cyclic way. So it's very similar workflow as the protein one. Once you're done with your RNAi, you can move on to the protein and then merge those 2 data onto the same slide. Again, now it's an actual product. You can have a lot of information on our website. We also have our experts here today. So if you want to discuss this directly, you can pass by our booth and have a chat about it. Also for the Maxima, similar to the ultramicroscope, we have our own workflow, of course. You don't just get the Maxima as a fully automated platform to perform your staining and imaging. We also have a bunch of sample carriers, depending on which tissue types we are working with. Of course, we have pre-tested reagents. This could be either in the form of off-the-shelf antibodies or already tested protein RNA panels, which are very helpful, especially for those customers who do not have too much experience, are not really sure on how to set up their panels. So we made it for you and made it even easier. Software is key when talking about spatial biology, because the amount of data that you're going to get out of even one experiment is massive. And if you don't have a clear pipeline on how to handle those data, it's going to get overwhelming. We have our own software, the microscopy view analysis software, which really helps you on each step and sets up a workflow for you to make sure that you really get the data that you were asking for. Also in this case, of course, scientific and technical support. Now, if you think about what I just said regarding our products, each of our portfolios has its pros and cons, right? You have 3D imaging that allows you to get an overview of your sample on a limited number of markers. And on the other side, you have spatial biology with the Maxima that allows to go for as many markers as you need, really. There's no limit to that, but on a single tissue slide. The nice thing about it is that they are kind of complementary though, right? So what we thought is, what if we would be able to develop a workflow that allows you to combine the 2 technology? In this way, you could look at 3D, have a complete overview of your samples for those 2-3 markers of interest, identify the region where you really want to go in depth with your analysis, make a slice out of it, and then perform the spatial biology application. So now you're really doing meaningful analysis on the exact region where you want it to. And this is quite a cliffhanger, but it's the moment where I'm going to hand over to Harold, who's going to show you exactly how he did it with his experiment. I'm looking forward to your talk, Harold. If you have questions, I'm happy to take them after the presentation or later at the booth. Thank you.
Hello, everybody. Thank you, Luigi, for the nice introduction. So, I'm a neurobiologist. I work in neural development. My whole career, I worked on understanding how the neural system is set up in a normal situation, how the brain is created from stem cells, how stem cells behave, how they can be manipulated, etc. And it's only very recent, two years about, that we got interested and kind of pushed by technical approaches into this field of cancer biology, at which we cannot avoid looking at it from a developmental angle. It's what I am. And I will show you a couple of examples and approaches towards this. So, okay. Here we go. Okay. So, stem cells in the mammalian brain, the subventricular zone. It's now recognized, accepted that all our brains contain and maintain stem cells throughout life, and that they, in theory, maybe practically can make new neurons, and in any way they can make new glial cells. So in humans, the use of these cells is debated, not their existence. But in mice, and that was the model I used for the last 30 years, sorry, I guess that's me, that I used for the last 30 years has been very well characterized. So in both species, we find clusters of cells along the walls of the lateral ventricles. So this, actually I can't see the pointer, so you have to believe me. In the walls of the lateral ventricles, which contain astrocyte-like cells, these blue cells here, the red cells, blue cells here too, in both cases, in human and in mice. And these cells are mainly silent, but can be activated and produce neurons, astrocytes, oligodendrocytes, etc. So, do you see the pointer? Yes, you see. Okay, so what happens in mice? Let's focus on mice. That's what we work on. I don't know much about humans. So in mice, we have the walls of the lateral ventricles produce permanently neurons that integrate into a structure called the olfactory bulb, this very rostral structure, which is a relay for olfactory information from the nose to the brain. Okay, and almost 15 years ago, a PhD student in my lab developed a technology, electroporation of postnatal animals, where she injects DNA solution, like for example a GFP-containing solution, and transfects this by an electric pulse and targets a couple of these stem cells in the wall of the lateral ventricle. This would be the ventricle, and these are individual stem cells, and we talk about the other cells that you see in a second. And with this method, you can label the entire system. So the cells here that are generated and the result of this generation in the olfactory bulb. Okay, and this is just what the system looks like. It allows you to recognize, or to look at this at high resolution. So we have initially stem cells that we target with our electroporation. At 8 hours after this electroporation process, they still have the shape of radioglia. 1 day later, 1 DPE, individual cells are generated. These cells enter in proliferation. At 2 days, there's a huge amount of progenitors, of neuronal precursors that is produced. These cells start migrating into the rostral migratory stream in a process called chain migration, go into the olfactory bulb. Here they separate from this mainstream and migrate individually into the target layers, where they become these red and blue neurons that you see on this section that have different morphologies. Okay, and over all these times, we were interested in the molecular regulation of this process, identifying microRNAs, transcription factors like Zik proteins in the stem cell compartment, factors that regulate migration like NCAM or CXCR4 and 7 in the migration process, relin, which is this detachment signal telling cells to go individually, and so on. NeuroD, for example, that we identified together with Miltony 15 years ago. It was Olaf, who should be here, showing that NeuroD1 is expressed by these cells and is needed to induce the terminal differentiation. Okay, that's all about, I will say, about development. Now let's switch to cancer. It's now recognized and well known that the human subventricular zone contains cells of origin for glioblastoma. It's not clear if it's the only cells of origin, but at least there are cells that can give rise to glioblastoma. And it's also clear now that in mice, subventricular zone neural stem cells can serve as cells of origin for glioblastoma. So based on this and our possibility to electroporate these cells, we developed a method it's based on 2 vectors up here. One vector is so we transfect these vectors by vivo electroporation. One vector is a transitory vector, leads to the knockout of P10 and P53 and P53. And at the same time, via the piggyback system, we integrate the EGF receptor into targeted cells. So a permanent integration. And with the EGF receptor, we integrate mCherry, so we know which cells express a knockout for EGFR and P10 and express the EGF receptor. And of course, we were expecting to get tumors, and that's what we get. Here you see tumor progressions from 1 week after electroporation, 2 weeks, and 4 weeks. It's a rapid and controlled process. Okay, we asked histologists. We didn't know anything about cancer, so we needed to be sure that we are looking at something that looks like cancer. So we asked specialists, Aurélie Chongandian and Professor Dominique Figarella-Branger at the hospital, to look at brain sections, use a histopathological approach to look into our tumor tissue in mice. We show basically all characteristics of glioblastoma, migrating cells, here cells migrating in the corpus callosum. We see necrotic palisades, we see overproliferation here by Ki67, and massive expression of OLIC2, and many other criteria. Basically, every criteria that has been described as an histopathological criterion for glioblastoma is fulfilled in these mice. These tumors kill the animal. It's a very rapid process. It starts at about 35 days after electroporation, and at about 50 days, all animals are dead under standard conditions. On the other hand, if you express only the GF receptor or control mice, we don't see any effect of electroporation. So it's a relatively cool model. It has surely disadvantages, but it has many advantages. For example, there is no need for transgenic mice. This you can do in whatever knockout mutant you have. You add CRE to the system, and you can look at a developing cancer in the absence of whatever gene you have a conditional mutant for. And you don't need to do major breedings. You don't have to wait for a year to have the right line. You just do the experiment, which is a major advantage. It's precise. There's a precise starting time point. We know when things start, and I will show you why that can be important. And it has a known cell of origin. These green cells along the wall of the lateral ventricles. Okay, so it's a relatively controlled information, which was for us one of the main criteria in the whole thing. So normally, and so far, we look at tumors in 2D, 2 dimensions, seeing growth here in histological sections at 1, 2, or 4 weeks over time. But of course, tumors develop in 3D. It's not just a section, a surface. It's a 3D structure, a highly complex structure. So we try to get a look at this development of tumors in all the dimensions, in time and in space. So Marie-Catherine Tiveron and Lena Vilvendre in my group, they devised the protocol. There is the electroporated animals at P0, and then looked at brains, isolated brains, at 1, 2, 3, and 4 weeks after electroporation, and then used the Blaze microscope to directly, in contact with a cubic cleaning protocol, which allows to look directly at fluorescence. Okay, so in this context, we look at the cherry fluorescence with a Blaze microscope over time. So that's what we get. So at 1 week after electroporation, at 0.0, we don't see anything. We checked that actually. At 1 week, we see small groups of cells along the walls of the lateral ventricles. So the ventricle is like a surface. Here you can see, you look into the ventricle, and you see small groups, small clusters of M-cherry immunofluorescence along the walls of the ventricles. After 2 weeks, already the growth is massive. At 2 weeks, the whole surface of the ventricle is covered. Individual groups of cells detach from the main tumor, and the tumor is huge. This gets worse, of course, at 3 weeks. Well, almost half of the hemisphere is filled up with the tumor. You have individual cells spreading away, but I'm pretty sure that some cells are caught up by the growing tumor, so we don't really know that. We're looking into this. And at 4 weeks, almost the entire hemisphere is full with this tumor, and and it's basically where we need to cull the animals to avoid suffering. Okay, so, and this is a highly reproducible thing because we can quantify the growth of these tumors now in large series. So here we look at 6 animals at each time point. At 1 week, 2 weeks, 3 weeks, 4 weeks, it's a log scale. So you see that growth is massive, and you can see how close these groups are together. So it's a relatively simple experiment. You can treat lots of brains. So for quantification, for example, for looking at therapeutic effects, it's a very interesting strategy because it's relatively little work because you don't need to do all the sectioning. And it's relatively precise because in a given experiment, the error margin is very, very low. So I showed you that at 7 days, we can observe these little clusters. Of course, we have to look from afar with light sheet microscope. So can we, based on this observation that we have already probably growing structures at these early time points, can we study these very early steps? As I said, I'm a developmental neurobiologist. I'm interested in these early events leading to a cellular process. So this is work from Nathalie Corré and Iliana Fontana in my group that looked at high resolution at these during the first week of the tumor development process. So at 2 days, the upper lane, basically not much happens. We have these electroporated cells. So here we express GFP, not nuclear M. cherry, but cytoplasmic GFP to see the entire cell surface or the entire cell filled up. So at 2 days, not much happens. We have many cells in the subventricular zone here, and very few site outside, and there is not much overproliferation, which is shown on the right. At 4 days, situation changes already, because we see now large amounts of cells that exit the subventricular zone and go to the corpus callosum, so this axon tract overlying the subventricular zone, and the overlying cortex, which is quantified here. And we also see that cells that leave now the subventricular zone, that should be here, they start to overproliferate. There we have the fraction of EDU or positive cells in the GFP positive fraction is massively increased. And at 7 days, that's what we see. So we have a massive accumulation of those such clusters in the axon tract, the corpus callosum, and individual cells that spread out from this structure in the overlying cortex. So tumor cells, induced tumor cells, undergo an early invasion step. It's not something that comes up when the tumor is big and cells start emanating. They start doing this at a very early time point, 1 week after transformation. Okay. So what I showed you so far is data on localization, so in this 3D and in 2D data, but a bit from a look from afar. It doesn't tell us anything about what happens to the cells, what are the molecular mechanisms behind this process. We know about tumor size, about shape, about localization and invasion. But we want to go deeper, and then in the discussion with Andreas Bosio, who is here, we decided to maybe have a deeper look into the molecular constellation, the molecular composition of these tumors, the molecular properties, the cellular composition, and the environment surrounding these tumors. Okay, so we used mixed technology. I don't have to introduce that. It's been done by Luigi in detail. And this was a close collaboration between the Miltini team, especially Fabio and Silvia, and in my group, Hugo and Marie-Catherine Tiveron, who were responsible for this part of the project. So what do you do in such a case? So you take a brain, you induce a tumor, exactly what we did before. Here we used, that's what you see. Then you make brain sections, lots of brain sections, and then you select a single section based on what you see on a binocular as it contains fluorescent cells. And that's what we sent to Miltini for analysis. This is the brain section one of the brain sections that we analyzed, and that I will focus on during the next couple of minutes. So this is what you see, just to orient you. So this is the lateral ventricle. This surface here is where we electroporated the transforming vectors. 2 weeks later, the tumor is spread out. We'll come in detail to different aspects like migration and etc. But basically, we select more or less randomly a section that contains the tumor and look at the tumor at high resolution. And we learn a lot. So this is a an overlay of 5 different markers. CD146, it's hard to see, but it's in blue. It's blood vessels, a blood vessel marker. CD11B, the immune system in the tumor, tumor-associated macrophages. Nestin is a marker for the tumor itself. N labels the neurons surrounding the tumor. And neurofilament labels the axons that come from these neurons or from other neurons, other brain parts. Okay, so close look at the tumor itself, different components of the tumor, and its environment. So we segmented this section, so this is the case for this specific section. We identified about 6,000, I can't read it from here, 6,000 something tumor cells and some almost 14,000 non-tumoral cells based on a set of markers. Here, for example, you can see that. So the violet population here expresses tyrosine hydroxylase, N, different myelin markers like MOG or I can. So basically, this population expresses all markers that worked that are non-tumoral but represent subpopulation of substructures in the normal nervous system. And then the other clusters are smaller. They all express, it's the lowermost lane, M-cherry. It's a criterion. You can discriminate easily the tumor from non-tumoral cells and express to different degrees the other markers that we found to be expressed, and we go through this. For example, the tumor, the environment expresses, shown here in green in the upper lane, N. They are all excluded from the M-cherry positive tumor, and GFAP, nestin, and GFAP are expressed in the tumor itself, which is represented in this list of in this heat map. So, and already at that level, if you look at the distribution of these different clusters, you see that the tumor is somehow structured. Not all cells in the tumor are randomly intermingled. There are green cells that are more in the center. There is part of the cluster of the tumor, for example, here, which doesn't contain this green cluster. We don't know what that means for the moment. Also, the list of markers is relatively short. But it's in agreement with the fact that a glioblastoma is a structured, I read that in one paper, a malignant organism which is highly structured in itself. Okay, so now we can look into processes linked to cancer development. Angiogenesis. We have here 4 markers that have been linked to angiogenesis. CD105 and CD146 are both expressed in the tumor, so you can already see this huge amount of new blood vessels that the tumor generated and massive angiogenesis. It's much higher than outside of the tumor that you see here. CD45 and CD46 are both expressed in and out of the tumor, but we have also markers like CD34, which are more or less specific for the tumor blood vessels, which is shown here in the quantification. And for example, EphA2, an ephrin receptor, which has been linked to angiogenesis, is expressed only in the tumor and not outside this tumor. So that already gives us a hint to communication, molecular communication between the tumor and its environment. It's no proof for nothing, but it's an entry door into studying these processes. And this is just higher magnifications showing the excellent staining on the one hand. On the other hand, also showing here that, for example, ephrin A4 is a diffuse staining coming from the tumor, while here CD34 and CD105 are highly confined to the blood vessels within the tumor. So it's one of the entry points into studying this process at a high resolution in a single experiment. The immune system, the tumor immune system, a big issue is half of the talks in this meeting deal with the immune system fighting the tumor. Okay, so we have 4 markers here in our list of 40 markers, CD45, CD11B, both markers for basically all tumor-associated macrophages. CD169 has been described and shown to be a marker for cells that are derived from the bone marrow. Okay, so cells immigrating the brain under inflammation and cancer conditions from the bone marrow. And CD11C, which has been described as a population of microglia inducing this marker as a population. And this is just 2 cells here one expressing CD169, one expressing and CD11B, one expressing CD11C, you can't really see that, and CD11B next to each other and next to a tumor cell in the vicinity of the main tumor. So we can look very closely into the entire complement of tumor-associated macrophages, again, in the same experiments we were looking for at before. Okay, so what about the environment? The tumor is not alone. The tumor is surrounded by neurons, here in green. The tumor is red. And, of course, the axons coming out of this tumor were generated by these tumors, here in blue. So already at this magnification, if you look at the neurons standing in the middle, you see that this layer here, so what you can see and what we see and what interests my colleagues that are mechanobiologists is that the tumor exerts pressure on the environment. You can see that these neurons are dense, very dense, close to the tumor. The tumor pushes these cells together and squeezes them, and there is quite an effort now to understand the mechanics of tumor biology in the brain, and some of my colleagues started to work on this question. Okay, so let's go deeper into neurons, just some observations. So we see cells emanating from the tumor, migrating in, invading cells from the tumor margin, which is about here, and going into the tissue through these existing neurons, the red cells individually. There are some surviving cells in the tumor, for example here, on the left side, close to the ventricular surface. There are still some surviving neurons. And when we go further away from the core of the tumor, we see that there are actually quite a few surviving neurons here on the right side that make it. So it seems as the tumor, on the one hand, pushes the tissue apart, on the other hand, engulfs neurons over time, and we imagine that these cells die over time in the tumor. It's a kind of gradient that develops over time. So there is an interaction between these cells at various levels. And there's an interesting point, so where are these cells going to? Because these cells that you can see here, there is kind of a gap between the tumor and this group of cells that gets out of the tumor. And that's an interesting observation. So here we combine the same labeling for the tumor in red with neurofilament. So tumor cells, it has been shown in many studies, tumor cells use axon bundles as a way of, as a pathway form for invasion. And we can see that these cells accumulate around these axon bundles, which are the so-called striosome, big axon bundles in the brain. So they seem to accumulate. So this here I added, by simple click in the program, EGF receptor as an additional marker, which gives us a nice outline of not only the nucleus by M. Cherry, but the entire cell as it expresses high amounts of the EGF receptor. Okay, so we can look at both things. On the one hand, position of the cells, and we can quantify this easily at the level of M. Cherry, and at the structure and orientation of these cells using a cellular marker like the EGF receptor, specific for the tumor cells. Very nice for analysis. Okay, another pathway that has been described in many studies for invasion of glioblastoma cells in man is the corpus callosum, so this structure here that connects both brain hemispheres. It's big bundles of myelinated axons. And when we look into this bundle here at the upper part of the tumor, we see that actually this neurofilament, so axon bundle, is used by tumor cells to invade the other side of the brain, basically. Okay, it's a very clear observation. So from a single overlay of structure, now of course you can add markers and markers and markers. They are all there, and ask with which molecular components, with proteins, this process is correlated. And that's what we do. Okay, so this is the last migration slide that I'm going to show you. No, it's not almost. So another migration, there is a periventricular migration that we observed. So we see here this group of neurons, of tumor cells in the dorsal part of the brain. There is this group in the ventral part, and we believe that this group migrated along the ventricle into this ventral aspect to settle down and generate a secondary tumor. So, and to finish this up, is another way, another mode of migration of tumor cells that has been described is vascular, using the vascular system for invasion. And so you're now used to seeing this section, M-cherry positive cells. These are that express the EGF receptor. And at some point, what struck us was this group of cells. So these two groups of cells that I just showed you. I can't see the pointer, that's the problem. Okay, so these two groups of cells, which are linked, appear to be linked with blood vessels, major arteria that pass through this brain region, the septum. I blow this up and it becomes clearer. So this we're looking now at this region at high magnification, so the tumor cells expressing here M-cherry and the EGF receptor, surrounding a blood vessel expressing CD105. And actually they are riding on, it looks like they are riding on this blood vessel and going far away from the tumor in a very efficient way. And that's an interesting observation that came up of these studies, that one of these blood vessels shows large amounts of CD169 positive cells. You remember? It's the bone marrow-derived macrophages, here in blue. So already at this early stage of immigration, we see the immune system reacting to invading tumor cells. Okay, so this is great. I just showed you a glimpse of all the things we get. And as Luigi said, it's so easy to get lost. As a neuroanatomist, I started working with a program and looked at it, and it's extremely hard to get tough information because there's so much out there, too much there. So we have to focus on a couple of things. But of course, we get a deep insight into cellular composition, molecular properties, and environment by such a simple experiment. Simple experiment. Okay, but you know what I mean. Let's get more complicated. But of course, the situation is more complicated because we are not dealing with a brain section, a tumor which is two-dimensional. We are dealing with a tumor that is three-dimensional. Okay, so this is a tumor exactly of the same age, which is far more complex. And all we see is here one single section at about this level. So of course, it would be really cool, and that was already introduced, to combine these both approaches. To have first a look from afar, look at the tumor in all its dimensions, and then go into it and ask what are the molecular components, what is the regulation, what are the different parts of the tumors doing. Okay, and this has been addressed. Well, we were lucky because Kevin came to us, so Kevin from Miltini, together with Victoria and Fabio, but Kevin was the driving force behind this project, trying to find a way to combine, to do first 3D imaging, and then on the same tissue with the knowledge that you have before, do molecular and spatial proteomics analysis. So, and that is the workflow that he designed. So, but using our mouse model, electroporation model, we decided to use it at 2 weeks. We fixed this tissue. There is permeabilization, immune staining, dehydration, clearing of the thing, and then 3D imaging. Then these the images would, of course, be analyzed. Where is the tumor? How is it organized? What is the directionality? Identifying actually the major axis through this tumor, giving us the most information about a given tumor in space and time. And now, in the next step, based on this information from 3D, define an axis for sectioning and do the mix analysis. And that's what he did. So this is now based on the Miltini clearing protocol, an experiment done on the expression of GFP. It's in red, but basically what we express is GFP. It's just the antibody, the nanobody that Kevin used to visualize the tumor. So we can see now the tumor, and it's of high quality. We can follow the tumor over in all its dimensions. We can follow individual clusters emanating from the tumor. You can imagine the ventricular surface along the tumor wall. It's as of good quality as we see with the cubic protocol when we look at direct immunofluorescence. Okay, so that is cool. Now we have information, the information we need. We know how the tumor grows. We can do this over time, and we know where we are. So the next step is, of course, to analyze this data and define the major plane of section, which is, of course, not the simple plan that we used before, going through the brain coronally and having one random section. No, what he wanted to do is to look into one particular plane which contains the maximum of the tumor on a single section. So that is here. And then and that was basically Victoria and Kevin, developed an approach based on a preformed agarose block that allows to section this particular brain in an angle of 41 degrees. So they calculated if we cut it at 41 degrees, we get straight through the tumor at exactly the level we want. And of course, that was following by sectioning. So now this is on the left side, you see a section, the light sheet projection section of this tumor. On the right side, you see a 10 micron section through this light sheet section. Exactly the right area, so that worked nicely. You can see all the components. It's very clear that he hit the right plane. Okay, and now, of course, he processed this section or these sections by mix and used something like, I don't know, over 100 markers. And for 15, we got a really interesting and nice signal. Okay, so this is a list of signals. We see tumor markers like, of course, the EGF receptor, CD44, several integrins, integrin beta-2 and beta-1, GFAP and nestin. We see GLAST and EphA2, so proteins expressed by the tumor itself. We see tumor-associated cell populations like CD45, so tumor-associated macrophages, ICAM1, which is an inducer or a chemoattractant for these macrophages when it's shedded. We have a marker that worked for the blood vessels, CD146. I already talked about that. And an interesting marker, we will come back to this, which is CD271 or P75. It's a low affinity NGF receptor, which has been described in glioblastoma. And interesting one, so this specific one is the only one that worked after light sheet, but not before. So there is some variability. But of course, many of the antibodies didn't work in the light sheet material, but worked on the normal mix. Okay, but 15 is quite a lot. We were surprised. It was really spectacular. So the quality is outstanding. So this is material that has been before analyzed by light sheet microscopy. So here we have a set of markers. We have nestin as tumor marker, CD271 as a marker that we will talk about in a second, CD146 for blood vessels, nu-N for the neurons, and neurofilament. You are used to this now. So the quality of these sections is as good as it is coming out of the normal, of the mix-only approach. Okay, this is another, just a proof of quality. Here we look at two markers that we used in both settings, GFAP and nestin. On the left side, after mix only, it's quite good and very informative, but it looks even better after the light sheet microscopy. We have many other cases where it's a bit less good. So there is some variability, but for GFAP, for example, and nestin, the quality is outstanding and the tissue is not at all damaged due to the light sheet transportation process. Okay, so now we can do what we did before, look at the specific processes. For example, what's happening in the immune system? We have information for ICAM1, CD54, which when it is shedded attracts immune cells, tumor-associated macrophages, and CD44, which is a marker for these TAMs. Okay, and we can see that in different regions now, regions that we know, we studied before, region 1, 2, and 3, there's a correlation between the amount of ICAM1 that we can detect in the tissue. The core is 1, the periphery is 2, and the very periphery is 3. So the higher the level of ICAM1, the more CD45 cells we can observe in this region. Makes sense, is an entry point into the study of this, of the immune system. Vascularization, we have the only marker that worked here was CD146, a perfect marker. We can study now vascularization based on the expression of tumor markers like of tumor markers like CD44 or non-tumor markers like NUN for neurons, the interface between the tumor and the normal environment, having markers at hand that works in both situations, 3D, that works after 3D analysis. So we know where we are exactly at the right level. So the last point I want to make is the point on CD271, the low affinity NGF receptor, which is expressed, there is data that it's expressed in brain tumor stem cells. It's not very clear, but there is some solid data. And it's expressed on the highly infiltrating cells, which are, as far as I understand, the major, maybe the biggest problem in glioblastoma therapy, this rapid emigration of cells from the main tumor. So it's supposed to be expressed in both populations. So how is it expressed in our situation here? So we looked into two predefined areas, C1 and area 1 and area 2. And so the tumor marker we use is CD44. And you see that CD271 is highly expressed in a very small population in the center of the tumor. It's highly variant, so it's in the core tumor, the amount of these cells is quite high. In the periphery, here, area 2, it's quite low. So this is coherent, at least to my eyes, with the existence or with an expression in a subpopulation. We don't have a proliferation marker in experiments, so it would be nice to see if these are low proliferating cells within the tumor, to see if that could be cancer stem cells. But at least it gives a good impression, a good insight, and good way to look at such specific markers. And then I want to make a last point on invasion, so the second function of CD271. And that was something which is quite interesting because it's a nice way to correlate the 2D and the 3D data. So if you look at here where the arrow is, there's a small population of cells which is not connected to the main tumor. Can you? Hello? Now go back. Just switch on. Yes. Okay. So this population of cells, you can see that in 3D. It's this little population. Well, the pointer doesn't work nicely, but basically it's where this arrow is, and you can detect it. You can see it in the 3D projection too. So we know where we are. It's a population which segregated, is not connected anymore to the main tumor. It's this little population. So what is expressed in this cell population? What makes it interesting? So it expresses the EGFR. That is normal. We express the EGFR. It's the oncogene. These cells express CD44, though they are tumor cells. They're transformed cells. Maintain CD44 expression. They express CD271, the low affinity NGF receptor linked to migration of these cells. And that is also interesting, they're actually in a bundle of axons. So they're using, as I suggested before, based on you remember that, on the 2D alone data, these axons, these cells are strictly confined to axon bundles, using them as highways to hell. Okay, and this is just another last point. It's the same cluster of cells, just overlaid with the staining for GFAP, and we can see that already at this early stage of migration, immune cells, we believe that these are GFA-positive activated microglia cells, already migrate towards this group of cells. In the tumor, they are highly present, but already this group of cells is recognized as and obviously attacked by the immune system. Okay, I think I stop here. Well, I don't think so. I will stop here. So I hope for me, that was a whole new world. Looking at not only at 3 and 2, also the 2D data was spectacular, very easy, and I couldn't stop working on this and trying to understand and make correlations. The 3D, 2D is something that works now. Okay, we start with 15, but a little bit of work, and this will be a very usable method. We should also not forget that most of the antibodies we use are raised against human tissue, so only very few cross-react. Okay, so it will be very easy, I would say, to detect a panel of antibodies which works in both conditions, and I think that's something that Miltony will do. The quality is super. I think I've never seen such a thing before. I think it's the first time that this really worked, and I'm glad that we were chosen as the beta tester for this new approach. And well, I think I stop here. I'm already too long. Sorry about that. So this work has been done to a large part at Miltenyi, to a large part in my group in Marseille. The key people are Kevin Bigot from Miltenyi, Victoria and Fabio, who did the experimental work under the supervision of Melanie and Sylvia. In my group, the work was mainly done by Marie-Catherine Tiveran, Nathalie Corré, Hugo, who left the group now, and Lena Villevendre. And we are collaborating with clinicians at the Marseille La Timone Hospital. And that's all I wanted to say. Thank you for your attention.